Hello, fellow biologists. Welcome to the first lecture of what will be several lectures about how we get to plants and eventually what plants do. At this point, I believe that Dr. Stevens has set an excellent background for all of you guys. So let me just start by quickly reminding you of where we are. And so as I go through my lectures, what I'll do is I'll try to start with a phylogenetic tree, and I'll try to start with a you are here indication. So if you've ever seen the t-shirts with the big galaxy, this is the Milky Way, and then a tiny little dot that sort of points to Earth and says, you are here. So that's what I'm going to try to do with these trees. I'm going to try to start by introducing us to each topic with sort of a you are here moment. Where are we in this grand phylogenetic tree of life? So as Dr. Stevens showed you, this is sort of our first big tree that encompasses all living organisms. And he spent a little bit of time talking about the bacteria and the archaea, and then what makes a eukaryotic cell a eukaryotic cell. And so, of course, this third domain over here is the eukarya, all organisms that have a eukaryotic cell. And there are lots of characteristics, of course, that make a eukaryotic cell, including the fact that they have chromosomes and multiple chromosomes, and they have a nucleus, and lots of membrane-bound organelles. But in particular, I'd like to remind you that all eukaryotes have mitochondria. All right, so those are the organelles that perform aerobic cellular respiration, and some of them have chloroplasts. And it's this latter group of eukaryotes that we're going to particularly look at in this lecture, right? Those eukaryotes that have chloroplasts. And so this gets us to the concept of what we're going to talk about today, which is algae. And so algae is a term that I'm sure you've heard used before. It's not a taxonomically distinct term. It's not like plants, or it's not like animals. There are a number of different organisms that can go in algae, and they don't all have a common ancestor. But they do have some physical characteristics in common. And so often when we talk about algae, we're talking about cells that are eukaryotic. Of course, they're photosynthetic and they're aquatic. They live in water, and they can be either unicellular or multicellular. So in this lecture, we're going to talk about the algae. And to get to algae, we have to first figure out how we get chloroplasts. So we're going to talk a little bit about the evolution of chloroplasts. Dr. Stevens mentioned that in his previous lecture. So once we have the evolution of a chloroplast, and we figure out how we get chloroplasts, then I'll very briefly mention unicellular algae, and because this is a course on multicellular organisms, I'm not going to spend a lot of time there, so this is just going to be a show and tell, but I'll point out a few groups of unicellular algae. So, for example, we have several different types of algae over here. A couple of these are unicellular. One is multicellular. We have some more unicellular algae over here. So two other groups of unicellular algae here. And of course, our red and green algae are multicelled. So I'll talk about just sort of do a quick show and tell of some of these unicelled algae, and then I'll spend the bulk of time talking about multicellular algae. And so there are three major phyla of multicelled algae. The Phaeophyta, which is this group right here, the brown algae. You can see that they're somewhat distantly related to our other two groups, which are going to be the red algae, which are the rhodophytes, and the green algae, which are the chlorophytes. And as we'll see, it's the green algae that probably gave rise to our modern land plants, what we consider to be our plant kingdom. All right, so to get into the idea of what an alga is, we need to think about how we get a chloroplast. And so to figure out how we get a chloroplast, we have to remember a little bit about endosymbiosis theory. And so as you recall from Dr. Stevens' lecture, the idea behind endosymbiosis is that we get particular organelles, especially mitochondria and chloroplasts, through this idea of a larger prokaryotic cell engulfing a larger eukaryotic cell engulfing a smaller prokaryotic cell. And so if we have a larger eukaryotic cell over here and engulfs some sort of bacterium over here, then this bacterium, if it does not get digested, will remain inside the cell and can even reproduce inside the cell, and this will become a mitochondrion. All right, so for all eukaryotes, there seems to be best evidence suggests, based on molecular evidence, there's a single origin of mitochondria. And so why do we think that mitochondria originated by this process of endosymbiosis? And there's a whole lot of evidence. Again, Dr. Stevens mentioned some of this evidence, but I'd like to sort of mention two key pieces of evidence. One is that if we look inside our mitochondria, it has DNA. Unlike just about any other organelle except for the nucleus. And the mitochondria have DNA, and this DNA seems to be configured like bacterial DNA. In other words, we don't have sort of these long strands of chromosomes, these single chromosomes. We have circular DNA. And the second point is that there are multiple membranes. There are pair membranes, at least in mitochondria. And so the idea is that one membrane represents the membrane of the prokaryotic cell that was originally engulfed, and the second membrane over here represents the membrane of the eukaryotic cell that engulfed it and then eventually turned it into some sort of vacuole-ish like structure. Okay, now if all eukaryotes share a single mitochondrial ancestor, how about chloroplasts? And it seems that there's also a single origin for chloroplasts, although it's going to get a bit trickier as we're about to mention. All right, so again, what's our evidence for chloroplasts originating via endosymbiosis? And again, we see DNA from bacteria, specifically DNA from what seems to be a cyanobacterial ancestor. And we see a couple of membranes from this cyanobacterium, and then other membranes that can be associated with the eukaryotic cell. All right, so very similar to the evidence that we have for the origin of mitochondria. Okay, so here's our little picture from your textbook figure of some sort of cyanobacterial ancestor being engulfed by a larger eukaryotic cell, and now we have a structure that we would refer to as a chloroplast that can multiply inside the host cell, and of course provides the host cell with carbohydrates from photosynthesis. Okay, now this single origin of chloroplasts denotes a group of organisms, and this is a taxonomically rigorous group. These are the archaeoplastids. So plastid, of course, for chloroplast, archaeo refers to ancient. And we have two groups of algae that are descendants of this, direct descendants of this archaeoplastid ancestor. We have the red algae and we have the green algae. Oops, all right. But what about all these other groups of algae, like the brown algae and a lot of these single-celled algas? Didn't we say that they also had their origin from this single evolutionary event? Sort of. And that gets us to the idea of secondary endosymbiosis. So I'll move up here so I can talk about secondary endosymbiosis. And so the idea behind secondary endosymbiosis is that a eukaryote without a chloroplast engulfs another eukaryote, not a cyanobacterial cell, but another eukaryote that already has a chloroplast. And it seems this has happened at least a few times. So here's how it might work. So here is our original origin of chloroplast. So here we have a eukaryotic cell, here we have a cyanobacterium. It gets engulfed, it becomes a chloroplast, a membrane is lost over evolutionary time, and then we get diversification into our two major archaeoplastic groups, the red algas and the green algas. Okay, and you can see inside of each one we have a chloroplast. So here's the chloroplast inside the red alga, here's the chloroplast inside the green alga. All right, now what happens though if this red alga is engulfed by another eukaryotic cell? So here is an even larger eukaryotic cell. We get this process of secondary endosymbiosis where now our chloroplast in our eukaryotic cell actually has its origins as another eukaryotic cell with a chloroplast. All right, and so it seems that this has happened at least a few times. So first of all, it's happened once with the red alga. So here's our red alga ancestor, secondary endosymbiosis, giving rise to our straminopiles, and our straminopiles will include brown algas. We'll mention those in a second. Also our alveolates, and I'll just use one example of an alveolate. They're single-celled, mostly single-celled algas. I'll use dinoflagellates as an example of an alveolate in just a moment. It also seems to have happened at least a couple of times for our green algae. So here are two endosymbiosis events over here, one giving rise to euglenids, which are sort of single-celled swimming, photosynthesizing algal cells, and then also our chlorarachnophytes over here. And I guess they have a chloroplast, and the arachno is because they have some sort of pseudopoda over here that look sort of like a spider, I guess, if you use your imagination. All right, so why do we think that this is going on? Well, our evidence is that we have plastids now. Instead of just having a couple of membranes that are from cyanobacteria, we now have 4 membranes, right? So a couple that are from the cyanobacteria, one now that is from the eukaryotic cell that held the cyanobacterium, and then another from the secondary endosymbiont that engulfed the original, engulfed the eukaryote. So we're up to 4 instead of our 2 from the cyanobacteria. And not only that, but this cell over here that gets engulfed now should have DNA not only from the original cyanobacterium, but also from the original eukaryote cell. And in fact, we even see in these examples over here a reduced nucleus in some of these secondary chloroplasts. So yes, it seems that secondary endosymbiosis has happened at least at least 3 times, according to your textbook. All right, so just to show you then what this is going to look like on our grand tree of life, I've cut out this bottom part over here. So we focus just on the eukaryotes. And so this group over here is our archaeoplastids, right? So here's where we get our original endosymbiotic origin of chloroplasts, giving rise to our red algae and our green algae. All right, so over here I'm going to abbreviate this archaeoplastids over here. Okay, then through secondary endosymbiosis, we give rise to a couple of other groups over here, the straminopiles and the alveolates. Also through secondary endosymbiosis, we give rise to a couple of different single-celled algae over here, and the cercozoans that had the archaeoplasts, and also the excavates over here, which had the our other group of green algae, or green alga descendants. All right, so that's what we mean over here when we have a sort of a single origin of chloroplasts originating by endosymbiosis. That's what we're really talking about. We're talking about one group of archaeoplastids, and then we're talking about these secondary endosymbiotic events. Okay, so we've got chloroplasts now. So I need to move again. So hello from this side of the screen. So what I'm going to do is I'm going to talk about unicellular algae, and then I'm going to talk about multicellular algae. And as I said, with the unicellular algae, there are lots of different groups of unicellular algae, but unicellular algae are not quite the focus of our course on multicellular organisms. So I'm just going to do a quick show and tell on a couple of really cool groups of unicellular algae over here. One is the dinoflagellates, and the other are the diatoms. So the dinoflagellates are an example of alveolates, and the diatoms over here are an example of our straminopiles, and they are at least straminopiles with our brown algae, which we're getting to in a second. So the key thing to note about these, both dinoflagellates and diatoms, where did their chloroplasts come from? Their chloroplasts seem to be derived from secondary endosymbiosis from our red algae and green algae over here, our archaeoplastids, but particularly the red algae. All right. Oops. Moving again. So about dinoflagellates. Dinoflagellates, of course, as we said, are single-celled algae. They're free-living, mostly marine, and the di part in dino refers to not the dinosaur, but to they have 2 grooves. Right, so you can sort of see a couple of grooves here, a groove here, and another groove down here. You got a groove here, right? So we see these grooves over here, and dinoflagellates have a couple of grooves, all right, and they have flagella that stick in the grooves, and so the flagella kind of wrap around the dinoflagellates like this. So this is what dinoflagellates do. Okay. Now, there are lots of dinoflagellates in the world, but just to give you an example of some that might be relevant ecologically and economically. One very particular important group of free-living dinoflagellates happens to cause what's known as red tide. And so if they become overabundant in certain marine environments over here, the tide can actually turn red. Remember that they're evolved from a red alga, so we sort of get a red coloring over here. And if they become, again, too abundant, they can actually produce a toxin. They can infect a variety of organisms that are harvested for seafood, clams, mussels, etc., and actually do a whole lot of economic damage to fisheries. So that's one example of dinoflagellates. But a more positive example from our perspective of dinoflagellates are a group that are symbiotic and actually live within corals. And these symbiotic dinoflagellates are known as zooxanthellae. So here's a little coral polyp, and coral polyp is actually sort of an entire animal, and corals are colonial animals, as we'll learn about later in the semester. And corals are capable of catching their own food with their little tentacles over here, and they stuff it in a mouth, and they have a digestive cavity right here where they digest their food. But they also rely fairly extensively, in fact, usually for over half of their energy, on these symbiotic algae. And so that is in fact what makes coral reefs such productive ecosystems, is the fact that corals have these dinoflagellate algae. So what I'd like to do, and I'll do this for this slide and at several points throughout the semester, is I'd like to show you just a little bit of a video clip that illustrates what zooxanthellae are. So it's one thing to learn about the taxonomy of these organisms and the structure of these organisms, but I also want you to have appreciation for what these organisms are doing in our world, and especially since we might not have the opportunity to take field trips to places like the aquarium or the zoo or the Crone Conservatory, where we can actually see more of these organisms. So I'd like to show you just a little bit of a video clip about zooxanthellae.
Corals are small animals, but they build structures that you can actually see from space. As we zoom in, we see the reefs where I work, on a little island called Ofu in American Samoa. The ridge there, the edge, is the coral reef, and as we zoom in, we see what those reefs are. Now, the reefs are made of individual coral colonies, like we see now. That coral, then, is actually made up of a whole series of small polyps, they're called. This is a colonial organism, and the polyps themselves have all the structures that an animal needs. It has a mouth, it has tentacles, it has gonads, it can live, it can reproduce, it can grow. They're in a colony of genetically identical polyps. Now, the color on these tentacles, like I said, is not the color of the coral itself. It's the color of the symbiont. And as the focus racks in and out a little bit here, then what we see is that we can just see the little globules of the symbiont. Well, let's take a closer look. We'll go into a tentacle and see those. These cells, the symbionts, are not just floating around. They're actually inside the coral cells. Corals are simple. They just have two cell layers, an epidermis and a gastrodermis inside. The symbionts are inside the gastrodermis, and you can see it there. Now, this is a life form called a dinoflagellate. It has chloroplasts because it's photosynthetic, but it has very odd-shaped chloroplasts, like these yellow structures here. We're going to zoom in to the chloroplast itself because that's where the damage happens during bleaching. What do chloroplasts have in them? They have membranes called thylakoid membranes. Those membranes hold the proteins called the photosystems that then capture light energy and turn it into chemical energy. It's the molecules that turn all of the sunlight that we get on the planet into the food that we eat. The rain of photons down here hits these photosystems and they gather them up. Now, if the temperature goes up and if the light goes up, then they freak out. There's too much energy, the photosystems break, and they no longer can function the way they do. But the rain of photon keeps going. the energy is still there, and as a consequence, that energy is now turned into reactive oxygen molecules. Those are damaging to cells, so it damages the inside of the symbiont, it damages the inside of the coral cell, and they spit the symbiont out. That spitting of the symbiont out by one coral cell isn't bad, but if the entire colony does it, then that's coral bleaching. What you can see here is simulated of the spitting out of these symbionts and the gradual whitening of this particular part of this particular coral colony. Well, when that happens across an entire colony, then the coral turns from its normal tan color into a white color. What difference does that make? The symbiont provides 75 to 80% of the energy the coral needs to survive, and without that energy, it can't make a skeleton and it can't live very long. So as a consequence, a lot of the corals that bleach eventually die.
Second example of a unicellular alga would be diatoms. And diatoms can be both in freshwater and marine environments. They're very important in marine environments as a source of phytoplankton, but so they have an ecological value, but they also sort of have a really interesting value in terms of helping us understand a little bit more about our climate, particularly our past climate. And so the idea behind a diatom, the di refers to the fact that it sort of has these two halves, like these two sort of silica plates that fit sort of nestle inside each other, almost like petri dishes. But these petri dishes aren't all just uniform and round. Notice that these two plates can take on a variety of different forms based on the species of the diatom that we're talking about. So not only do they have these very distinctive shapes and very distinctive morphologies for each species, but the composition of diatoms can change substantially based on the conditions of the aquatic environment, based on things like temperature and salinity and nutrients, etc. And so if you do a core, if you collect a core in the sediments of lakes or of the ocean floor or something like that, and you look at the diatoms that have accumulated at different stages in the geological past, what you can see is that the climate is changing for particular environments. And so especially since the last glaciation about 20,000 years or so ago, or since the most recent period of large glaciation 20,000 years ago, diatoms species compositions have fluctuated in freshwater bodies all over the world. And so we get a really good idea of how climate is changing from our diatoms. So that's sort of a little fun fact about diatoms. All right, so I'll move again, and this takes us over to multicellular algae. And multicellular algae are commonly referred to as seaweeds. All right, so if you go to the beach or anything like that and you see sort of a large clumpy something, large bodied organism that's an algae that's sort of moving around in the water or maybe washing up onto the sand, you call it a seaweed. It probably belongs to one of these three major groups. So we have our brown algae, or our phaeophytes, and these are straminopiles like our diatoms. And then we have our red alga and our green alga, and our red algas and green algas are archaeoplastids, and in green algas in particular, as we're about to see, are probably a group that gave rise to our modern land plants. All right. So let's start with our brown algas, our phaeophyta. And one example of large brown algas would be kelps over here. And so if you've ever seen a kelp, and here's a picture, sort of a drawing of a kelp, here's an actual photograph of some kelp, one of the things that you start to notice about a kelp is that it seems to look kind of plant-like. But the key is it's not plant-like. So what makes it look plant-like? Well, it's got these things over here, right here, that sort of look leaf-like, and they are the blades. It also has things that look kind of stem-like, and those are called the stipes. So notice that we've got some stipes going on in here. Okay, and then it has some things that look sort of root-like, but they're not roots. They do anchor the alga though, and these are referred to as holdfasts. So we have at least some morphological similarity. These are analogous structures, convergent evolution, but they're not the same as stems and leaves and roots. And finally, we have a structure over, a lot of kelps have structures that aren't analogous to anything that we have in plants, and those would be floats over here. So here you see an example of the float, and the float helps the plant, particularly sort of the stipe and a blade over here, sort of remain buoyant in the water and sort of flat so that it can collect light and photosynthesize. Okay, so why aren't these stems and leaves and roots? And the idea is that these structures over here, blades, stipes, and holdfasts, lack transport tissue. And we're going to talk a little bit more about what the transport tissues are in plants, but these don't have any real transport tissue. And so the question is, why is transport irrelevant? And we're going to talk a little bit more about that, but think about where these brown algae are living. They're living in the water, all right? And so any cell anywhere in this body of the alga that needs water, well, it's got it. It's surrounded by water. Any one of these cells that needs a gas like carbon dioxide, well, the gas is dissolved in the water, so it's got it. It doesn't have to move the gases around, which means that all these structures can photosynthesize fairly well. And finally, nutrients. You know, we think of things like nitrogen, phosphate, etc., right, that plants need. Again, plants have to take it from the soil up into the leaves and the stem. Well, again, these nutrients are in the water. They're surrounding these kelps. So transport is not nearly the need in large multicellular algae that it would be in plants. We don't have transport tissue. All right, but what can we say, though, about brown algae and what makes them similar to other algae and what makes them a little bit different from other multicellular algae? And so let's take a look at the chlorophylls. So in brown algae, and in fact, in all algae, we have chlorophyll A. All right, and so that's a particular chlorophyll molecule that existed in this first cyanobacterium ancestor, probably, that was engulfed and became a chloroplast in our archaeoplastids, right? So our archaeoplastids have chlorophyll A, red algae do, green algae do, and of course brown algae do too. Brown algae, though, also have chlorophyll C. All right, so chlorophyll C is probably something that evolved after this secondary symbiosis event, or at least that's one hypothesis. Okay, finally, again, I wanted to mention a little bit about the ecology and the economic value of these brown algae. And so they're very common, particularly in marine habitats, particularly in temperate ecosystems. And for all of the excitement that goes around a coral reef and all the research that is being done and the concern that's being directed towards coral reefs, there are also other very, very productive marine ecosystems on our planet in the temperate waters. And a lot of these temperate marine ecosystems have brown algae, these giant kelp forests, which are really spectacular ecosystems in their own right. And we'll see a little bit of a video on that in just a second. A quick note about some economic value. A number of cultures, particularly in Japan and Southeast Asia, rely heavily on brown algal species for food sources. Laminarias in particular are great food sources, very nutritious. And finally, even we here in the West, we're not eating usually laminarias in our soup, but we do distill an algin, that we use as a thickener for a lot of Western food. So starches, so a lot of stuff like ice creams and so on that sort of have a thickener comes from this algin over here. So whether you intend to or not, you've probably eaten some brown algae in your life. All right, so let me quickly show you a little bit of a video about these extraordinary kelp forests that can exist in temperate waters and in particular off the coast of the Northwest in the United States.
Kelp forests can be seen along much of the west coast of North America. Kelp are actually large brown algae that live in cool, relatively shallow waters close to the shore. They grow in dense groupings, much like a forest on land. These underwater towers of kelp provide food and shelter for thousands of fish, invertebrate, and marine mammal species. Kelp are very simple organisms that consist of a holdfast, a stipe, and blades. At the bottom is a root-like structure called a holdfast that anchors kelp to rocks and other materials on the ocean floor. Young kelp must compete for space to settle and grow, as the rocky bottom is carpeted with smaller algae and invertebrates like anemones and sponges. The stipe is similar to a plant's stem. It is strong, yet flexible, allowing kelp to sway in the currents of the ocean. Many fish use this middle area of the kelp forest as hunting grounds. The blades contain a special gas which act like a float, keeping the kelp blades close to the surface of the water where they absorb energy from the sun. This ability to float raises the kelp to the sea surface, forming a dense canopy. The canopy serves as a nursery or brood area for many organisms due to the warmer surface temperatures and slower water currents. Many organisms, from small fish to birds and even whales, use the thick blades as a safe shelter for their young from predators or even rough storms. Kelp forests have a greater variety and higher diversity of plants and animals than almost any other ocean community. NOAA's scientists study kelp forests by visiting the same locations over and over to assess the presence and abundance of a variety of organisms. Monitoring allows marine scientists to determine if the kelp forest is changing over time and to identify the cause of those changes, whether natural or human.
Do you like that little look at a kelp forest? I hope you got your moment of zen from seeing a very happy seal frolicking in the kelp forest. If you ever get the chance to frolic in a kelp forest in your life, I encourage you to do it. They're amazing places. All right, now let's take a look at our next major group of multicellular algae, seaweeds, and we're going to go into the archaeoplastids. And so not only are we going into the archaeoplastids here, we're also going into a new phylogenetic tree. We're zooming in a little bit. So I'll move over here so you can see the tree. And what we've got now is sort of a condensed version, tree 2, that focuses in particular on the archaeoplastids. So here's the archaeoplastids over here. We have our rhodophytes, our red algae. We have our green algae over here, or this group right here. And of course, then that is going to give rise to our plants. So let's start with our first group, which would be the red algae. So this is the red algae, our rhodophytes. And again, there's morphologically, they do look superficially similar to plants, and I won't say a whole lot more about that since you have the basic idea of blades, stipes, and holdfasts now. But let's think briefly about their photosynthetic pigments. And so they do have chlorophyll A, like all of our algae, right? So chlorophyll A originates with our archaeoplastids over here. However, they don't have chlorophyll C, like brown algae. Instead, what they have is phycourethrin. And so this is a red pigment that allows them to capture different wavelengths of light, particularly wavelengths of light that are more in the blue and the green wavelengths, which are the wavelengths that penetrate deeper into water. And so the really interesting things about red algae is that it's, not the color is not just sort of a nice feature. The color really determines its ecology. And so there are red algae that can live closer to the surface of the ocean, but there are also some red algae that can get really deep, over 200 meters at depths. There are still some red algae, and there's just not a lot of light down there, but they're still doing photosynthesis at this very low level over here. So red algae are very ecologically important because they can live at so many different, they're species that can live at so many different water depths. They're also economically important. If you like sushi, nori is this red algae seaweed that goes around sushi, so that's good. Okay. Which takes us to our green algae. So again, they're archaeoplastids over here, but this is our group of green algae. And so phylogenetically, what we notice about green algae is they're not what we refer to as a monophyletic group. They don't all have a single common ancestor that unites the entire group, unlike our red algas over here. They do have, however, a diversity of forms, sort of a real diversity of forms, going along with the fact that they're taxonomically a fairly diverse group. We have some that are actually colonial organisms as opposed to multicellular organisms we see with this zygnema over here. We have some that are truly multicellular, and so here's an ova, and you can see that its body is differentiated with specialized structures. You see these very sort of leaf-like blades over here. And then finally, we sort of have these very odd green algas that are sort of lack distinct cell dividers, but they do have lots and lots of nuclei, and they still sort of produce these structures that sort of resemble superficially leaves and stems or rhizomes or things like that. So a lot of morphological diversity over here in our green alga group, which is about to be important really quickly. Right. In addition to this diversity in morphology, let's think about their photosynthesis. And so, as we've said, they have chlorophyll A, like all of our archaeoplastids, but they also have chlorophyll B. And so chlorophyll B absorbs a slightly different set of wavelengths, and in particular, it gives these algae sort of that more darker, sort of vibrant green color that we associate not just with green algae, but with plants. And so it turns out, right, that our green algae, make a note over here, our green algae have chlorophyll B, which distinguishes them from the red algae, the brown algae, and other algae. All right, so for that reason, among others, because they have chlorophyll B, it's thought that the green algae are, of all the algae, those that are most closely related to our plants. Also, there's something that's really important about the ecology of green algae. I'll move so I can show you the different sorts of habitats in which green algae can live. So, of course, our chlorophytes can live in marine environments and some freshwater environments. So here's a little sea lettuce over here living in a very shallow tide pool in a marine environment. But the really interesting thing is that there are some species of green algae that can even sort of be terrestrial, land living. So notice the sort of the reddish color over here. So this is not some hiker on a glacier slowly bleeding out. This is what's actually known as, these are actually algae. This is what's known as watermelon snow. And so there are green algae that can live in the snow at high elevations. They do produce the secondary red pigment to sort of help protect them from UV radiation, but in fact they are green algae. They've got chlorophyll B, and there can even be some green algae that live on the sides of buildings. So green algae are sort of starting to make this transition from being aquatic organisms, which is how we define all algae, to being a bit more terrestrial, which is really cool. All right, so we've got our green algae, our chlorophytes broadly, but let's zoom in and let's take a look at some specific groups of green algae. In particular, let's take a look at a class of green algae called our charophyceans, charophycea. And so the really interesting thing about charophycea is that they produce a particular chemical called sporopollenin. And so sporopollenin is a chemical that is a very resistant chemical, particularly for dehydration, but also to all sorts of other stresses. I mean, you can actually put sporopollenin in algae and boil the acid and boil the acid, and sporopollenin will resist boiling acid for a certain period of time. So it's a very resistant polymer. And so this very resistant polymer is unique over here, not to all green algae, but to our chlorophytes. Sorry, but to our charophyceans. So here's where we get sporopollenin. All right, so notice that separates this group from other groups. And so this is a figure 29.4 in your textbook. Take a look at it if you want to. However, you see that even within our charophyceans, there are other groups over here. All right, and so it seems that some charophyceans are particularly closely related to plants, and those would be three genera: Coleochaetes, Chara, from which the charophyceans get their name, and more recently it seems like maybe Zygnema might also belong to this group as well. And so these are the charophyceans that are particularly closely related to land plants. And so what makes these charophyceans particularly related to land plants? And it seems that one of the key features that at least some of these charophyceans have, particularly Coleochaetes and Chara, is they have a life cycle that is closely related to land plants. Now, we haven't said a lot about algal life cycles, and that's because algae, these multicellular algae, these unicellular algae too for that matter, have a real diversity of life cycles. And in some multicellular algae, as Dr. Stevens mentioned, you can see algae that have a multicellular diploid, a 2-end phase, and a multicellular haploid phase. You can see some that have a multicellular haploid phase only, others that have a multicellular diploid phase only. All right, but Chara is going to have a few life cycle features that seem to sort of set up the types of life cycles that we're going to see in plants. So let's take a look at those. Oh, but first, we need to take a quick look at what these genera look like. So here is a modern coleochaete picture. You can see that it's a group of cells sort of in a disc. They can be about the size of a quarter or a little bit bigger. And they have fossil ancestors that go back at least 415 million years. So this is still, of course, after land plants evolved, but it at least indicates that they have very old ancestors. So here is Parka, a fossil genus of a green alga that looks very much like modern coleochaetes. And then here is Chara, and we have Charan fossil ancestors that are over 410 million years old. And here's what our modern Chara looks like. And so notice that our modern Chara is clearly a very multicellular organism with very differentiated structures, stem-like structures, leaf-like structures, very particular reproductive structures, as we're about to see in a moment. So let's take a look at the Charan life cycle. And before I get too far into the Charan life cycle, what I'd like to do is just sort of review a life cycle with you, and let's start with the animal life cycle. This is an animal life cycle, and hopefully it looks fairly familiar to you. And so what we're going to do for all of our life cycles is we're going to start with a dotted line over here. So this dotted line is going to divide our life cycle into two parts. It's going to divide it into the diploid part, and I'll try to be consistent and put that on top, and the haploid part, and again I'll try to be consistent and put that on the bottom. And so remember, diploid cells are eukaryotic cells with 2 copies of each chromosome, whereas haploid cells are eukaryotic cells with just 1 copy of each chromosome. And so for most animals, like this happy little worm over here, this annelid, this earthworm, with its anatomically incorrect smiley face, these multicellular organisms are diploid, right? So the worm is a multicellular diploid organism. We're multicellular diploid organisms, and that's more or less true for all the animals that we know. Now, how do we go from a diploid cell to a haploid cell? Well, at least for any organism, that happens through meiosis. So meiosis is the process of taking one diploid cell, going through meiosis, and we get 4 daughter cells, but these daughter cells are haploid. All right, so I've shown a couple of different meiosis events. So one meiosis event produces 4 haploid cells that are eggs over here. Another meiotic event produces 4 haploid cells that are sperm over here. And together we would refer to these, the eggs and the sperm, as gametes. And so what you've always learned, or mostly learned in your previous classes, is that whenever meiosis occurs, we get gametes. And that is, of course, true for animals. But as we'll discover in a little bit, it's not true for a lot of other organisms. So what happens then to these gametes? Well, of course, the way it's supposed to work is that a sperm cell fertilizes an egg cell. So here we have a fertilization event, and that takes us from a 1-end cell and a 1-end cell. They're now fused. That gives us a 2-end cell again. So we're back across the line over here. We have a 2-end cell, which is a zygote. And then, of course, our zygote and subsequent cells go through a whole lot of mitosis until we have regenerated our multicellular diploid organism, our worm again. All right, so this is the basic animal life cycle. This is our Chara life cycle, and notice that our Chara life cycle is very different from our animal life cycle. In fact, it's almost flipped upside down. So for an animal, our multicellular generation was diploid, and we just had a bunch of single cells that were haploid. But for Chara, it's going to be the reverse. Our multicellular organism is going to be haploid, and our single cells over here are going to be diploid. So what is Chara doing? So Chara is producing a multicellular organism over here that is haploid. Then this multicellular organism is producing the gametes. It's producing sperm and it's producing eggs. Now, if you start with a haploid cell and you produce more haploid cells, these haploid cells can't be formed by meiosis. You can't take one set of chromosomes and divide it in half. Instead, these haploid cells have to be produced by mitosis, which is going to be a lot different than the way that you're used to thinking about where haploid cells come from. You're used to thinking about haploid cells coming from meiosis in an animal life cycle. Not going to be the case anymore over here with Chara, and in fact with a lot of plants and a number with the organisms that we're going to see. So mitosis produces our sperm cells, produces our egg cells. Again, if everything works as it should, a sperm cell fertilizes an egg cell. This is the fertilization event, which gives us a zygote. A zygote is just that one cell. At this point, what Chara is going to do is it's not going to go through mitosis. It's not going to produce this multicellular organism. It's just going to maybe let the zygote hang out for a little bit, but eventually, before the zygote divides by mitosis, it divides by meiosis, going from 2N back to 1N. And we have 4 cells that result from this meiotic event, and these 4 cells are referred to as spores. And eventually, or hopefully then, these spores, we'll take one of them for example, will divide a bunch of times by mitosis to return us to our multicellular haploid form of the organism. So we need a name now for this multicellular haploid form of the organism, because we're used to dealing with animals and we just call 1N cells gametes or sperm cells or egg cells, but now we have this multicellular form and we need a new name for it. So what are we going to call it? Well, Chara is sort of like a plant, so we're going to call it a plant. That's the phyte part over here. Now, what does this plant produce? Well, it produces gametes, so we're going to call it a gametophyte. And this is one of the, so a couple notes about how you start to read these botanical names that we're going to get, right? So a lot of them are derived from Greek and from Latin, and they sort of have a pattern whereby in order to really read them, you have to start from the right and read to the left, all right? So the rightmost part of the word is the noun, right? That's the phyte, the plant part, and then what comes before it is the modifier. So what is this plant doing? It's producing gametes. All right, and if you want your dumb bit of humor for the day, how do you remember that phyte refers to plants? Well, here's the silly joke. What do you call a group of plant biologists? They're the Phyte Club. Yeah, you saw that one coming. All right. So we've got our Chara life cycle, all right? However, our Chara life cycle, as interesting as it already is, has a few more twists. So let's take a look at these next twists. What we've seen so far in our Chara life cycle is summarized over here in this little life cycle series of events. We start with the gametophyte, which is haploid. Here's our gametophyte. It produces gametes by mitosis, which are also 1N. So here are the gametes, the sperm cell and the egg cell. Then, if we have fertilization, that gives us a zygote. So here's our zygote over here. And our zygote is coated in sporopollenin. And so remember, sporopollenin is this very resistant substance that charophyceans are able to produce. But now, the new thing about Chara in particular is that the zygote is actually covered in this sporopollen, which makes this zygote very resistant to things like desiccation, drying out, that might occur as you're moving from a from an aquatic environment to a terrestrial environment. Eventually, when our zygote is ready to undergo meiosis, it produces 4 spores, which are haploid again, and then one of these spores is divided by mitosis to give us our gametophyte, our plants that produce gametes. Now, if this were the chara life cycle, it would be interesting, but as I said, green algae have a huge diversity of life cycles. In fact, algae in general have a huge diversity of life cycles. This would just sort of be one life cycle out of many others, right? What makes this life cycle particularly interesting as we think about the evolution of plants, as we think about the transition from an alga to a terrestrial photosynthesizing organism? And they have really, really exciting gametophytes with gametangia. All right, so remember, so a gametangium over here, if we read our word, it's an angios, angium singular, angios, or angia plural. Gametangia is plural for containers. And what do these containers hold? Well, they hold gametes. And so we can have two different types of gametangia, or gamete containers. We can have an archegonium that has an egg and an antheridium that holds sperm cells. Okay. But again, what's so exciting about this? And the idea is that our archegonium is going to hold on to the egg until it's fertilized. And this might not seem like a big deal, but think about how all other algas have approached reproduction. Any other alga that's out there basically throws its sperm cells and throws its egg cells out into the water, and hopefully the sperm cells find the egg cells and fertilization happens, and then you've got a zygote and you're off and running again. But not chara. Chara actually is going to take care of its egg and take care of the kid, the zygote, so to speak, that comes with it. All right, so yes, chara is going to nurture its offspring. So what's going to happen is our sperm cells are going to leave the antheridium, the container that has sperm cells, and they're going to swim around over here and find the egg cell that's held in the archegonium. And they're going to fertilize that egg cell. And so I'll color it in to show it's fertilized now. And once it's fertilized, now it's that zygote. Now it's the cell that we have up here. All right, and so we're going to hold on to this egg cell, which is non-motile. We're going to protect it until it's fertilized and it can develop the spore pollen and coat of its own. That's going to allow it to resist all sorts of drying out or other challenges that might be associated with a terrestrial environment. All right, so this is really a big deal because this is going to allow us to take care of our zygote, take care of the products of fertilization. And as we think about the transition to land, one of the things that's going to become a problem is how do you do fertilization where a sperm cell is swimming to an egg cell in a world where you don't have water? And the answer is maybe you don't figure out that, but if you can figure out how to take better care of the products of fertilization, that is going to be a huge step forward, as we're about to see. So, what do these actually look like? Well, here is an archegonium over here with an egg. So here is an antheridium with lots of sperm cells, and the sperm cells will swim around and fertilize the egg. And hopefully in lab, you might even have a chance to see that, right? But if you don't, here's a quick little video to show you what it's going to look like. So here is the chara antheridium, and we have lovely guitar music to serenade our sperm as they are leaving the antheridium. So these little things right here are the sperm cells, and so you can start to see them swimming around here. There's another one down here. There you see one right here. So we're zooming in. There's one. There's one. There's one. And so on. All right, so let's cut to the chase. And this is fertilization. So this is just the opening of the archegonium. Here you can see the egg cell right here. You even see a little rotifer, but don't let that distract you. Look for the little sperm cell that's about to go swimming in and fertilize the egg. There it comes. And it goes. All right. Very good. Well done. So that's what fertilization looks like in a chara life cycle, and as mundane as that might seem, chara is one of the very, very few algae, particularly one of the very few green algae, that actually holds onto its egg and has fertilization inside some structure that contains the egg. What have we learned about the chara life cycle? Well, here are a few key features in review. First of all, the zygote is coated in sporopollenin, as we mentioned right here. And the egg and the zygote are protected in an archegonium, and particularly that is going to allow the plant, our gametophyte, to take care of this fertilized zygote until it can get the sporopollenin coat, until it's ready to survive some kind of desiccation event. And so these features, as we mentioned, are very, very advantageous, are going to be very advantageous in a terrestrial environment as we start to have a more difficult reproduction in a world without water. So, what I hope you'll be able to do at this point is to answer a bunch of questions. So these are some quick review questions for part 1. I will take a little bit of time and we'll cover the answer, go over the answers together as a class at the start of lab on Thursday. So please take a look at these questions. And also one other study opportunity for you is to take a look at this particular phylogeny that comes out of your textbook and label the key character states that are changing at each node. And so this will also get you to think about what's happening throughout the lecture. Notice that this is a slightly different tree than the one that we're presenting, Dr. Stevens and I, but if you can translate what you're seeing in that key to what you're seeing over here, that should be an indication that you're understanding the key character states that are evolving and making each particular group of organisms unique. Well, hope you've enjoyed the first lecture, and I look forward to seeing you shortly again in the second lecture when we'll talk about how plants have started to evolve to a terrestrial environment.